Hello everyone, I'm Dr. Kuhle, and today I will be teaching you about the neuronal membrane at rest. Today we're going to be talking about ions. These are electrically charged atoms that can hold either a positive or negative charge and contribute to electricity in nerve cells. We're going to talk about the different types of ions that exist inside and outside of the cell when it's at rest or when it's firing nerve impulses known as action potentials. We'll also be discussing the phospholipid bilayer, which is essentially a wall that lines the cells, and ion channels that exist in this phospholipid membrane. We're going to start off today's lecture by talking about the simple reflex. Here, we have sensory neurons that can send information up the spinal cord via the dorsal root to nerve cells called interneurons. Interneurons send signals to areas of the brain that register pain, such as the medial prefrontal cortex. So in red here, you can see the sensory neuron axon is projecting from the foot up to the body, and it is receiving the sensation that's painful after stepping on a thumbtack. Information is transmitted up the spinal cord via the dorsal root ganglia and back and up to the brain. Once that sensation is received in the brain, there are motor commands that are sent back down to the foot via motor neurons in the ventral root. As you can see here in blue, motor neurons are going to project down back to the bottom of the foot to allow our foot to move. And in this simple reflex, we are essentially protecting our body and commanding muscle movement. Motor neurons typically synapse at the neuromuscular junction, and they will synapse onto muscle cells that induce this simple reflex. So how do neurons send signals to one another? We know there are both electrical and chemical signals in the nervous system that allow neurons and other cells to communicate. But first, we're going to focus on electrical signals. Neurons use electrical signals that sweep along the axon. These are known as action potentials. So what induces these electrical signals in the cell? Well, it's electrically charged ions that exist in the cytosol of the axon. So how do neurons communicate electrically? Remember that neurons are bathed in salty extracellular fluid. So for example, here we have our neuron, and there's plentiful sodium around it. Sodium is a positively charged ion that helps to stimulate electricity. We have positively charged ions and negatively charged ions. For example, the two positively charged ions I want you to know for this class include sodium and potassium. There are also negatively charged anions, such as chloride and other organic anions. A special type of signal or nerve impulse is known as an action potential, and an action potential refers to the separation of electrical charge across the cell membrane. Here we have two different types of neurons. This first neuron here has a myelinated axon, and we can see that it's due to myelination, it has a thicker axon, a thicker diameter, due to extra layers of myelin. If you remember, myelin helps insulate the axon and maybe speed up these nerve impulses. If you remember, there are two different types of cells that create myelin. In the central nervous system, oligodendrocytes produce myelin, whereas in the peripheral nervous system, Schwann cells produce myelin. And so here, although this axon is myelinated, it's still not providing quite enough insulation, and thus the extracellular fluid that's around the cell is filled with positively charged sodium ions that help conduct electricity. So action potentials do not diminish over distance. They are fixed in size and duration, meaning that when a nerve impulse begins, it is not going to slow down or diminish. This is important because nerve impulses are how cells communicate. So, for example, an action potential or nerve impulse could start here at the axon hillock, and the nerve impulse will jump from node to node of the axon. These nodes are known as the nodes of Ranvier. So how do action potentials allow cells to communicate? We know that information is encoded in the frequency of action potentials of individual neurons, as well as the distribution and number of neurons firing action potentials in a given nerve. You may have heard the term cells that wire together fire together, and that is similar to this idea, in that certain areas of the nervous system have more nerve cells firing together. So information is encoded in this action potential, allowing cells to communicate. We can go back to the simple reflex as a great example of this, in which nerve cells will fire to induce a reflex at the neuromuscular junction and command the muscles in our foot to move from a sharp object. The action in action potentials occurs at the cellular membrane, as shown here. The entire neuron has a cell membrane that's composed of something called a phospholipid bilayer. In this cell membrane is where action potentials occur. Cells that are capable of generating these nerve impulses or action potentials include nerve and muscle cells. Nerve and muscle cells both have an excitable membrane in which ions can help generate electricity to induce these nerve impulses. There are certain things you should know about the neuronal membrane. First, the electrical charge of the neuron at rest, how electrical charge can be rapidly distributed across the cellular membrane during the action potential. Remember, this is when a nerve impulse is firing and the cell is not at rest, or how the impulse can propagate reliably along the axon. Remember, it does not diminish in strength or frequency over time. Just like humans, neurons need to rest. When this neuron is at rest, it cannot fire a nerve impulse or action potential. If a neuron was to constantly fire these nerve impulses or action potentials, it could lead to detrimental effects such as excitotoxicity and cell death. We can think of the simple reflex example. We don't always want to cause muscle movements or twitching movements. Unfortunately, this is a symptom of many neurodegenerative diseases and disorders. So our neurons have to rest in between these nerve impulses so we're not constantly moving or exciting cells. Thus, cells need to rest in between the nerve impulses, known as action potentials. There are three main players that play a really important role in the resting membrane potential of a neuron. First, the salty fluids on either side of the cell membrane. So remember, that includes the extracellular fluid that bathes the neuron and the cytosol inside of the cell. The cell membrane itself, which is the phospholipid bilayer, and the protein that spans the membrane. We're going to talk about specialized channels and receptors that sit inside of the membrane and help regulate the amount of ions that come in and out of the cell. Each of these has properties that establish a resting potential. Water is the main ingredient of the fluid outside of the cell, which is the extracellular fluid, and inside of the cell, which is the cytosol. We have electrically charged atoms known as ions that are dissolved in water that play a role in neuronal rest and nerve impulses known as action potentials. Remember that water is a polar molecule. It has an uneven distribution of electrical charge. Here is the chemical formula for H2O. 2 hydrogen atoms and 1 oxygen atom are bonded together covalently. This means that they share electrons. However, an oxygen atom has greater affinity for these electrons, and as a result, the shared electrons spend more time associated with the oxygen atom than with the 2 hydrogen atoms. So oxygen develops this negative charge, whereas hydrogen develops this positive charge. The oxygen atom requires a net negative charge because it has extra electrons, and the hydrogen atom acquires a net positive charge. Thus, H2O is a polar molecule held together by polar covalent bonds. Atoms or molecules that have a net electrical charge are known as ions, which I previously described. We can take salt as an example. This is a positively charged ion, known as a cation. Salt is a crystal of sodium and chloride ions held together by the electrical attraction of oppositely charged atoms. Sodium is positively charged, whereas chloride, shown here in blue, is negatively charged. This attraction towards one another is called an ionic bond. Charged portions of water have a very strong attraction for sodium and chloride ions, and thus salt dissolves in water. The salt dissolves readily because the charged portions of the water molecule have this stronger attraction for both sodium and chloride than the ions have for each other. The electrical charge of an atom depends on the difference between its numbers of protons and electrons. Ions with a net positive charge are called cations, whereas ions with a negative charge are called anions. Remember that ions are the major charge carriers in the conduction of electricity in the neuron. These are the ions I'd like you to know for our class. The two positively charged cations we'll be discussing the most include sodium and potassium, and we'll also be talking about negatively charged anions such as chloride and organic anions. Here are the important ions that you should know for how the cell maintains resting membrane potential. When the neuron is at rest, there is more potassium inside the cell. As you can see here in gray, K+ is inside of the cell, whereas sodium, shown here in purple, and chloride, shown here in green, are more concentrated outside of the cell. Here is our neuron at rest. At rest, the inside of the neuron is actually more negatively charged than the outside of the neuron. We already know from my previous slides that the extracellular fluid contains plentiful sodium ions that are more highly concentrated outside of the cell. However, there's more potassium concentrated inside the cell. This might stump you because potassium has a positive charge. However, later on, we're going to talk about specialized channels that allow potassium to leak out of the cell and make the extracellular fluid more positive, in addition to plentiful organic anions that exist inside of the cytosol that play a role in the negative membrane potential of the neuron. So, why is the inside of the neuron negative at rest? We know that the cell membrane is actually more permeable to potassium ions, and thus they can actually leak out of holes in the cell wall and go outside of the neuron into the extracellular fluid. We're going to talk about these specialized channels that are selectively permeable to the potassium ion. Additionally, the membrane is only partially permeable to sodium, and thus there is less leakage of sodium inside of the neuron, maintaining a more positive charge outside of the cell in the extracellular fluid. There are also organic anions inside of the cell that contribute to the resting neuron's negative charge. All three of these play a key role in allowing the cell to rest in between nerve impulses that are known as action potentials. Let's take a closer look. So, here are the important ions for resting potential. As I stated previously, we know that the cell membrane is very permeable to potassium ions. Thus, there are specialized channels that allow potassium to exit the cell. Potassium leakage can lead to more positive charge outside of the cell, whereas the cellular membrane is partially permeable to sodium, meaning that more sodium is typically going to stay in this extracellular fluid, and there's less leakage of sodium inside of the cell at rest, leading to a negative charge inside of the neuron. However, when sodium leaks inside of the cell, this typically occurs during an action potential or nerve impulse. The cell needs to become depolarized or more positive inside in order for these nerve impulses to occur. But don't worry, we're going to talk about that more later when we cover action potentials in Chapter 4. So, what is this cell membrane made of? The cell membrane is composed of lipids. Lipids are water-insoluble biological molecules. And as you can see here, the lipids of the neuronal membrane form this barrier. It's essentially like a wall to water-soluble ions and to water itself. So how do neurons communicate electrically? Nerve cells and muscle cells have an excitable membrane that conduct these electrical signals. I want you to know a few key things about the phospholipid bilayer, which is the cell membrane. First, it has a hydrophilic head, meaning that it is water-loving. This is a polar molecule that borders the very outside of the cell by the extracellular fluid. It also has hydrophobic tails, which create a wall that protects the cell and prevents ions from just flowing in and out of it. Essentially, this phospholipid membrane creates a layer or wall of protection around the neuron. And the phospholipid membrane contains specialized proteins, not shown here in this picture, but we'll get to that later, that actually can sit inside of this wall and act as bridges for passages of ions in and out of the cell. Here's more on the phospholipid bilayer. Phospholipids have a polar phosphate group that's attached to one end of the molecule. So phospholipids have a polar head, as we noted before, that's hydrophilic, while the nonpolar tail is hydrophobic. So just to reinforce this idea, the phospholipid bilayer is essentially a wall that isolates the cytosol inside of the cell from extracellular fluid outside of the neuron. This contributes to resting potentials and action potentials. Proteins create receptors that are embedded in the phospholipid bilayer. What are these receptors? Well, these receptors are made of proteins, and they act like bridges for ions that can pass in and out of the cell. This helps regulate what can come inside of our neurons and what can leave them. In order to understand receptors, we want to understand how protein structure develops inside of the neuron. We have enzymes that catalyze chemical reactions. We have the cytoskeleton, which, if you remember, we've talked about microtubules and neurofilaments that help maintain the axon's shape. If you remember, microtubules also play a super important role in exoplasmic transport, allowing synaptic vesicles carried with important molecules or neurotransmitters to be transduced down the axon. We also know that receptors are sensitive to neurotransmitters, and this is why receptors play an important role in the phospholipid membrane. So, how are proteins made? If you remember, in Chapter 2, we covered the central dogma of biology, in which DNA is transcribed into messenger RNA, which has the ability to leave the nucleus. Messenger RNA goes to a free-floating ribosome or to ribosomes attached to the rough endoplasmic reticulum in order for protein synthesis to take place. Remember that the primary structure of a protein is simply a sequence of these small amino acids shown here in purple. They can come together to form a polypeptide chain. The sequence of protein is ultimately decided by the DNA of the gene that's encoded for that protein via this process. The change in DNA sequence can subsequently change the amino acid sequence of a protein. Even changing just one amino acid in a protein sequence can affect the protein's overall structure and function. Here is the basic structure of an amino acid. They all have a central carbon atom, which is covalently bonded to 4 molecular groups: a hydrogen atom, an amino group, a carboxyl group, and a variable group known as the side chain or R group. The reason I bring up R groups is they play a really important role. R groups are different on all amino acids because they help to create the structure or shape of a protein. The differences among amino acids result from differences in size and nature of these R groups. The properties of the side chain actually determine the chemical relationships in which each amino acid can participate. Essentially, the R group that's attached to the amino acids can help determine what structure or what shape the protein will end up taking on. So there are 4 levels of protein structure. During protein synthesis, we know that amino acids come together to form a polypeptide chain. This occurs during translation. There are 4 levels of protein structure. This is just the most basic one that we have discussed so far. So, as I previously noted, the primary structure of a protein is simply a chain of amino acids that are held together by peptide bonds. This is shown in step 1. This is the primary structure of a protein that occurs from translation. A secondary structure of protein means that as the molecule is being synthesized, the chain actually wraps up into this alpha helix shape. This is known as the secondary structure, shown here in step 2. Proteins can also take on a 3D structure, and this is known as the tertiary structure. Remember, R groups are a molecular group on amino acids, and they play a really important role in determining the shape or structure of a protein. So in this case, side chains will actually help develop the protein into this complex 3D structure. Interestingly, when these 3D structures, also known as subunits, come together, they can form a much larger molecule. So each polypeptide that contributes to the quaternary structure is known as a subunit. You can think of these subunits like little Lego blocks. When the Lego blocks come together to build a shape, this is the quaternary structure. Interestingly, subunits help to create receptors on the cell membrane. So these subunits, when they come together to form the quaternary structure, can become embedded in the phospholipid membrane of cells, and thus they can act as a bridge or rite of passage for ions coming in and out of the neuron. Ion channels are made from these membrane-spanning protein molecules known as subunits, and typically 4 to 6 subunits need to come together to form an ion channel that also forms this pore. And earlier we talked about how R groups or side chains help determine the structure or shape of a protein. They also help to determine the size or diameter of the pore. This helps a channel become selective to only one particular type of ion. It's important that these ion channels are selectively permeable to specific ions to help regulate homeostasis. Potassium channels are selectively permeable to potassium, and likewise sodium channels are permeable almost exclusively to sodium. And that's why the size of the pore can help determine what ions can come inside and outside of that channel. Remember that these proteins that are embedded in the actual phospholipid bilayer are ion channels, and they are selectively permeable to different ions that can come inside and outside of the cell. There are several different ion channels we'll discuss in this class. First, voltage-gated ion channels are controlled by membrane potential, and thus there are specific voltage-gated ion channels for particular ions, such as voltage-gated sodium channels, potassium channels, or calcium channels. There is an additional type of channel known as the ligand-gated ion channel, and this channel will open when a specific neurotransmitter binds to it. For example, as you can see here, when the neurotransmitter acetylcholine, shown here in red, binds to this receptor, it is going to cause the channel to open and allow ions to come in and out of the cell. Interestingly, there are 2 receptors we'll talk about, including the NMDA receptor and GABA receptor, that have ligand-gated ion channels. The NMDA receptor is specifically important because it's the main glutamate receptor in the brain, and it has both ligand-gated and voltage-gated ion channels, which we'll talk about later on. Additionally, GABA is the main inhibitory receptor in the brain. In addition to ion channels that are embedded in the phospholipid membrane, there are ion pumps, which are also membrane-spanning proteins that are existing in the phospholipid bilayer. They work simultaneously with ion channels to regulate the amount of ions that come in and out of the cell. Ion pumps work a little differently because they require the use of adenosine triphosphate, ATP, the main energy currency of cells. Ion pumps are actually enzymes, and their function is to break down ATP and use that energy to transport certain ions across the membrane. We'll see that these pumps play a critical role in neuronal signaling and transporting things like sodium, potassium, and calcium in and out of neurons. It's important for you to understand that ions can move one of 2 ways. They are influenced by different factors such as diffusion and electricity. Diffusion of ions occurs when the membrane potential has channels that are permeable to the ions, And later we'll talk about how certain ion channels are only permeable to particular types of ions, such as potassium. There is a concentration gradient in which the ions can flow in a particular direction. Now, I previously noted that water is the main ingredient of the extracellular fluid outside of the cell and the cytosol inside of the cell. Ions and molecules that are dissolved in water are in constant motion, and these electrically charged atoms are what create electricity in the body. Although ions typically do not pass through this phospholipid bilayer directly, remember, mainly due to these hydrophobic tails, diffusion causes ions to be pushed through these channels in the membrane. For example, ions can diffuse across the membrane utilizing two different types of transport, including passive transport, as shown here, when facilitated diffusion does not require the use of the energy source ATP, and via active transport, which requires ATP to be broken down as an energy source to pass ions in and out of the cell. First, let's talk about passive transport. So passive transport is the movement of molecules from an area of high concentration to an area of low concentration. So for example, if we were to drop dye in water, the dye molecules would be highly concentrated in this initial area. But once the dye molecules dissolve and mix into the water solution, they will try to go from an area of high concentration to low concentration to create a homogeneous solution. This difference in concentration between this low area and high area is called a concentration gradient, and ions flow down a concentration gradient. However, active transport works in an opposite fashion, as ions actually have to move against their concentration gradient rather than with it. For example, the cation sodium would move from an area of low sodium concentration to an area of high sodium concentration. This forces the ion to move against the gradient because a positive cation is usually attracted to a negative charge. Thus, this type of transport requires adenosine triphosphate. It requires this energy to push the ions against their gradient. The concentration gradient allows ions to diffuse across this phospholipid bilayer. So for example, if we look here, this is representing passive transport, in which ions are going to be moving with and along their concentration gradient. For example, potassium is going to move from an area of high concentration inside of the cell to an area of lower concentration outside of the cell. Whereas sodium is going to be attracted to this more negative resting membrane potential, and it's going to move from an area of high concentration outside of the neuron to an area of low concentration inside the neuron. To the right here, this is demonstrating the sodium-potassium pump, which is a specialized pump that utilizes adenosine triphosphate as an energy source to push sodium or potassium ions against their concentration gradient. So remember in Chapter 1, we learned how Ben Franklin's discovery of electricity helped scientists like Galvani and Emile Dubois-Reymond study electricity in the body. They quickly discovered that electrically stimulating cells at the neuromuscular junction caused muscles to flex. Now we're going to talk about how electricity works in those cells. So remember that ions are electrically charged particles, and you can induce a net movement of ions in a solution with an electrical field. We're going to look at figure 3.9 as an example. So if we had wires from the two terminals of a battery and they were placed in a solution of dissolved sodium chloride, we could look at the movement of ions influenced by this electrical field. Remember, opposite charges attract and like charges repel. There will be a net movement of sodium, shown here, towards the negative terminal, the cathode, and a net movement of chloride, an anion, towards a more positive terminal, or anode. The movement of electrical charge is called an electrical current, as you've probably heard before, and it's measured in units called amps. In this example here, positive current flows in the direction of sodium movement from the anode to the cathode. Opposites do attract, so you'll see that the ion concentrations are creating an electrostatic pull that is responsible for the movement of ions on an electrostatic gradient. As you can see, as we've talked about earlier, sodium is going to be attracted to the negative cathode, whereas chloride is going to be attracted to the positive anode. So how does current flow in neurons? First, there's electrical potential. This is the force that's exerted on a charged particle. So remember that voltage is represented by the symbol V, and it's measured in units called volts. More current will flow as the voltage is increased, and we're going to talk about that later on. Electrical conductance also helps current flow in neurons, and this is the relative ability of an electrical charge to migrate from one point to the next. And conductance depends on the actual number of ions or electrons that are available to carry the electrical charge. Driving an ion across the membrane electrically requires a few different things. First, the membrane must possess channels that are permeable to that ion to provide conductance. So for example, we could have ion channels that are permeable to potassium. 2, there is an electrical potential difference across the membrane. This difference is important because we need to have a balanced ratio of ions in and out of the cell to create this balanced electrical potential. A balance of negative and positive charge inside versus outside of the cell is required for a nerve cell to rest. How can we measure the voltage of the cell to determine if the cell is at rest or not? We can measure membrane potential, the voltage across the neuronal membrane at any moment. And I'm going to teach you how we can measure membrane potential in a neuron. Sometimes a neuron's voltage is at rest. At rest, a neuron is typically at a voltage of about negative 65 to negative 70 millivolts. Voltage can be measured by actually inserting a microelectrode into the cytosol of a cell. We'll take this example here in figure 3.11. You can actually measure the resting membrane potential in the cell by taking a cell in a petri dish and inserting a voltmeter into its cytosol in the cell soma. As you can see, the voltmeter can actually read if the neuron is at rest or not. The electrical charge inside of the neuron versus the outside of the neuron is measured in millivolts. The voltmeter will actually measure the electrical potential difference between the tip of this microelectrode and a wire placed outside of the cell. This method actually reveals that electrical charge is unevenly distributed across the neuronal membrane. The inside of the neuron membrane is negatively charged when the cell is at rest, whereas the outside of the cellular membrane is positively charged. How is membrane potential maintained? Well, remember that membrane potential refers to the difference in electrical potential across the cell membrane at rest. So if there's a difference in charge inside or outside of the cell, a nerve impulse can potentially occur. So how can we calculate membrane potential? The membrane potential is actually a weighted mean of the equilibrium potential of different ions. In this example, we'll use sodium and potassium. When the neuron is at rest, both sodium and potassium are able to cross the membrane. And we need to know the equilibrium potential for 3 key ions. The equilibrium potential for potassium is negative 80 millivolts, and the equilibrium potential for sodium is 62 millivolts, whereas chloride's equilibrium potential is about negative 70 millivolts. So what does this mean exactly? We know that the membrane potential is a weighted mean of the equilibrium potentials of the ions sodium and potassium. Essentially, sodium and potassium are in a tug of war. They're trying to maintain a membrane potential by going and moving in opposite directions. For example, sodium will try to move towards a more positive membrane potential. Let's look at figure 1 first. At a starting state, we have more sodium outside of the cell compared to inside of the cell. However, sodium ions are going to be attracted to moving towards a positive membrane potential, and thus some sodium will try to come inside of the cell. When intracellular sodium arises and moves in this more positive direction, they're moving towards their equilibrium potential. In the image on the right, we know that potassium is more concentrated inside of the cell rather than outside of the cell. However, potassium is going to be driving towards a more negative membrane potential and may efflux out of the cell. So thus, this tug of war between the two ions helps to maintain a membrane potential. And the membrane potential is the mean of the ion's equilibrium potential for sodium and potassium. Remember, sodium's is 62 millivolts, whereas potassium's is negative 80. Now let's focus on potassium for a moment. Potassium is special because there are more ion channels that are selectively permeable to potassium rather than sodium. And that's important because potassium is going to try to move the membrane potential towards its negative membrane potential. An efflux of potassium, in addition to organic anions inside of the cell, help the cell maintain its resting potential at about negative 65 to negative 70 millivolts. Remember, it's important for the cell to rest. So how does potassium regulate its equilibrium potential? Potassium will try to move the membrane potential towards its negative membrane potential. So thus, we know there's usually more potassium inside of the cell compared to outside of the cell. But if an ion channel that's selectively permeable to potassium is present, potassium is going to want to enter that channel. Next, if the potassium leaks outside of the cell, we know that this ensures that the cell is more positive on the outside and more negative on the inside. This is helping establish the resting potential in the neuron. There's less ion channels that are permeable to sodium, and this ensures that there is always more sodium outside of the cell. An equilibrium state is reached in which the diffusional and electrical forces are equal and opposite, and the net movement of potassium across the membrane ceases. This is balanced. As we can see here, the electrical potential difference that exactly balances an ionic concentration gradient is called an equilibrium potential. In this example, the equilibrium potential will be about negative 80 millivolts. And remember, the membrane potential of a neuron can be measured by taking the mean equilibrium potential of potassium and the mean equilibrium potential of sodium. So make sure you know this for your exam. You'll need to remember the equilibrium potential of these three ions. The resting potential of a typical neuron is around negative 65 millivolts to negative 70 millivolts. And this is actually a requirement for a healthy, normal functioning nervous system. Cells need to be able to rest in between action potentials, also known as nerve impulses. To understand the negative membrane potential, we need to look at the ions that are present and how they're actually distributed in and outside of the neuron. Here is the cell at rest. As you can see, there's more potassium ions present inside of the cell, in the cytoplasm, and more sodium ions present in the extracellular fluid outside of the cell. Here we have our phospholipid bilayer consisting of lipids that are helping protect the neuron and creating a wall. Remember that there are certain proteins that can be embedded in the phospholipid bilayer to allow ions to come in and out of the cell. These can be selectively permeable to a particular type of ion, like potassium or sodium. Also recognize that there are organic anions present inside of the cell, which also contribute to its negative charge at rest. So now we're going to move on to part 2 of the neuronal membrane at rest. As I said before, I'll usually post review questions in these slides, and you can challenge yourself at home and try to answer these questions to practice for your quizzes and exams. What guides sodium and potassium in and out of the cell to establish the membrane potential? Well, there are 2 important ion pumps we're going to talk about, including the sodium-potassium pump and the calcium pump. The sodium and potassium pump is very important because it regulates the ions that come inside and outside of the cell. It's working simultaneously with other ion channels within the phospholipid bilayer. It's very important that sodium and potassium are regulated because this could prevent excitotoxic cell death. Otherwise, the cellular membrane would become depolarized or positively charged too often, causing cells to fire and eventually die. This pump maintains ion balance in neurons by utilizing active transport to diffuse across the membrane. If you remember previously, we discussed that active transport requires an energy source. So this means that ions need to be pushed up against their concentration gradient. So remember, we've discussed passive transport, which is a form of diffusion in which ions move from an area of high concentration to an area of low concentration. Remember the example I gave with dye molecules being distributed evenly throughout water. The difference in concentration is called a concentration gradient, and in passive transport, ions flow down a concentration gradient. However, during active transport, ions need to move against their concentration gradient. And because they have to move against their concentration gradient, it requires the use of... During active transport, cells require the use of ATP. This helps them move against their concentration gradient. And the sodium-potassium pump uses ATP to allow ions to move in and out of the neuron. So here's the sodium-potassium pump. And this pump is working on overtime to make sure that we have the proper amount of sodium ions in and outside of the cell and the proper amount of potassium ions in and outside of the cell. So let's look at our example here. The sodium pump itself is an enzyme, and it can break down adenosine triphosphate and use its energy to help ions come in and out of the cell. So the chemical energy that's released by this reaction drives the pump and exchanges internal sodium for external potassium. The cytosol here is the inside of the cell, whereas up here this is extracellular fluid. The actions of this pump ensure that potassium is more concentrated inside of the cell, whereas sodium is more concentrated outside. Notice that the pump pushes ions against their concentration gradients. So let's take a closer look at the sodium-potassium pump. So here we have the sodium-potassium pump, and there are 3 internal sodium ions in the cell cytosol. ATP will come to the pump, and the pump itself is an enzyme, so it's going to break up ATP, and it's going to take one of its phosphate groups, shown here in orange. When the phosphate actually binds to the pump, it induces something called conformational change. It changes the actual shape of the pump and allows its gates to open. When the pump opens up, these 3 internal sodium ions are actually able to travel through it back out to the extracellular fluid. Interestingly, when these 3 ions reach the extracellular fluid, 2 potassium ions are able to come back through the channel. When the 2 potassium ions travel back inside of the cell, this dephosphorylates the ion pump. And so essentially, dephosphorylation ensures that the sodium-potassium pump goes back to its original shape. This is a cycle that continually occurs throughout the body to ensure that we have proper movement of these 3 sodium ions in the extracellular fluid and bringing back in these 2 potassium ions back into the cell. And this is driven by ATP. At home, you can watch this video by the Amoeba Sisters, which is a really cute representation of the sodium-potassium pump. You can go on and look on Canvas, where I've posted a link to watch this video to help you understand the sodium-potassium pump better. Also, I'm going to be posting some worksheets that you can use at home to practice drawing out the steps of the sodium-potassium pump. You can print these out or you can work on them on your iPad, but I'm going to walk through the steps with you so we can work together. Again, I've attached these worksheets to the module in Canvas so you can open this up and practice drawing out the sodium-potassium pump and labeling it at home. So remember that the sodium-potassium pump is utilizing active transport, in which it's trying to move molecules, known as ions, against a concentration gradient. Intrinsic carrier proteins called pumps are involved. They sit in the phospholipid bilayer and work simultaneously with ion channels. They utilize ATP to force ions against their concentration gradient. And this process is very selective because only specific ions can be moved through the pump. So here's our sodium-potassium pump. First, here's our phospholipid bilayer, which we've already talked about. Remember, we have our polar or hydrophilic heads and our hydrophobic tails that are constituting this bilayer. We also have intrinsic protein, which is the sodium-potassium pump itself that sits within the phospholipid bilayer. Here's adenosine triphosphate, the key energy source, and our potassium ions are shown here in black, whereas our sodium ions are shown here in white. You can label this on your worksheet. This pump will move sodium ions in one direction and potassium ions in the other in a 3:2 ratio. So let's go through each step. So let's go through all of these individual steps in the sodium-potassium pump. First, we know that this pump can be bound to ATP, which has a high affinity for the 3 sodium ions that are present inside of the cell. Remember that these 3 sodium ions were originally in the cytosol. However, this pump functions as an enzyme, so it can actually break down the energy source ATP, in which a phosphate group is donated to the sodium-potassium pump. When the pump is phosphorylated, it induces something called conformational change. And during this time, the pump can actually change its shape, which essentially means that its gates open. And once the gates open, those 3 sodium ions can actually move from the cytosol outside of the neuron to the extracellular fluid. The 3 sodium ions are now presented on the other side of the membrane in the extracellular fluid. Once these 3 sodium ions go back into the extracellular fluid, the potassium ions that are present, which are these 2 potassium ions shown here in black, can enter the sodium-potassium pump. This causes a dephosphorylation of the pump, meaning that phosphate group is removed, and when the potassium re-enters inside of the cell's cytosol, it actually induces a conformational change, and it goes back to its original shape. The pump now loses affinity for these potassium ions, and they're released back inside of the cell. So now ATP can bind to the pump, and this process repeats. So this is happening all of the time in your body. And remember, as long as ATP is present, the process can continue. And this is ensuring that we have the proper amount of sodium ions outside of the cell and the proper amount of potassium ions inside of it. Again, these worksheets will be posted for y'all on Canvas, so I definitely recommend printing them out and drawing out all of the steps and writing them in chronological order to help you practice for your exam, because you may have to label a sodium-potassium pump for your exam. Hint, hint. Another pump that plays an important role is the calcium pump, which is also an enzyme like the sodium and potassium pump, and it actively transports calcium out of the cell's cytosol across the cell membrane. Remember that ion pumps are working in the background to make sure that the ionic concentration gradients are established and maintained, essentially allowing the cell to rest or have an action potential occur. And without these pumps, the resting membrane potential would not exist and the brain wouldn't be able to. It's really crucial to regulate calcium in the cell. So this calcium pump is important for decreasing any intracellular calcium and maintaining a low level. These include intracellular calcium binding proteins and organelles like mitochondria, which typically sequester calcium ions. So why is it important to regulate calcium? Calcium plays an important role in a process known as exocytosis. So we're going to look at this image for me to explain this better. We've already talked about how synaptic vesicles function as small boats, essentially, and they can carry neurotransmitters or other important molecules from the top or axon hillock of the axon down the axon to the axon terminal at the synaptic cleft here. So remember, synaptic vesicles are carried by specialized proteins such as kinesin and dynein during exoplasmic transport. And during exoplasmic transport, those motor proteins are going to carry synaptic vesicles up and down the axon. And that's playing a really important role in getting neurotransmitters to the end of the synapse. At the end of the synapse, at the axon terminal here, calcium allows these synaptic vesicles filled with neurotransmitters to more easily get to the synapse. Thus, calcium triggers this synaptic vesicle exocytosis and helps to release neurotransmitters. If there's too much calcium that's present or produced in the mitochondria, it can lead to neuronal cell death such as Alzheimer's disease. Calcium accumulation can also occur following concussion. Also, I wanted to talk more about potassium channels. Now, I've already mentioned that there are more ion channels that are selectively permeable to potassium, and that's playing an important role in helping the cell maintain its resting membrane potential. For example, if there are more potassium channels available, there is availability of the potassium ions to go from inside of the cell to the outside of the cell. This helps the cell maintain its negative resting membrane potential, in addition to those organic anions that exist inside of the neuron. Most potassium channels have 4 subunits, and they're arranged like staves in a barrel and form a pore. Interestingly, this area called the pore loop is really important. The pore loop here functions as a selectivity filter, and it makes the channel more permeable to potassium. Interestingly, this pore loop was actually discovered by studying the effects of scorpion toxin, because scorpion toxin blocks potassium channels and poisons its victims by binding tightly with this channel pore. Studies in animal models have helped scientists understand the mutations involving only a single amino acid in the region and how it can severely disrupt neuronal function. For example, there's a strain of mice called the Weaver mouse, and these animals have difficulty maintaining posture and moving normally. They walk kind of imbalanced. And so this defect has been traced back to a single amino acid mutation in the pore loop of potassium channels. And this typically occurs in specific neurons in the cerebellum. If you remember, the cerebellum's at the back of the brain and plays an important role in motor coordination and movement. As a consequence of this mutation, sodium and potassium can pass through the channel, and increased sodium permeability causes the membrane potential to become less negative. Remember that the positive cation sodium, when it comes inside of the cell, is going to induce something called depolarization, which is going to increase the neuron's ability to fire action potentials, which we'll cover more in Chapter 4. However, this mutation disrupts neuronal function. The cell needs to rest in between these nerve impulses or action potentials. And in recent years, scientists have found that many neurological disorders, such as epilepsy, are explained by mutations of specific potassium channels. And this is because the cell requires this negative resting membrane potential in order to rest in between nerve impulses. Another consequence of high potassium permeability is that the membrane potential is particularly sensitive to changes in the concentration of extracellular potassium. So, for example, a change in membrane potential from the normal resting negative 65 millivolts to a less negative value is called depolarization. This is when there can be an influx of sodium ions inside of the cell and an efflux of potassium ions outside of the cell. Therefore, increasing extracellular potassium depolarizes neurons. So how does our brain regulate potassium? The blood-brain barrier can do a great job of limiting what comes in and out of the brain. It can also monitor the movement of potassium in addition to other blood-borne substances. In addition to the blood-brain barrier, glial cells like astrocytes can actually help regulate the amount of potassium. For example, they can take up extracellular potassium whenever the concentrations get too high to help regulate normal neural activity. Astrocytes actually have membrane potassium pumps that concentrate potassium in their cytosol. So thus, they have potassium channels of their own, just like neurons. And potassium enters the astrocyte through these potassium channels, causing the astrocyte's membrane to actually depolarize. So the entry of potassium increases the internal potassium concentration in astrocytes, which is believed to be dissipated over a large area by the extensive network of these astrocytic processes. If you remember in an earlier lecture, I talked about how astrocytes can perform something called glial cell buffering, in which they can eat up extra neurotransmitters that are in the synaptic cleft. Similarly, potassium spatial buffering allows astrocytes to eat up external potassium, and this can regulate the amount of potassium that's left in the synapse. Now that we understand more about potassium ions and sodium ions, I wanted to mention potassium chloride and give you a real life example. So you may have heard of Dr. Death before, and he was a doctor that was a supporter of euthanasia for people who were terminally ill. He, in fact, assisted many patients in ending their lives when they had terminal illnesses through the injection of potassium chloride. Of course, there's much controversy with this, as many people don't think that it is okay to end human life under any circumstances. So Dr. Death, what he did was he created this form of injection where essentially he would inject a patient with an intravenous line of harmless saline solution. When the terminally ill patient was ready to die, they would switch the solution to an anesthetic solution followed by potassium chloride. So essentially, the anesthetic would cause the person to become unconscious and suppress the activity of neurons in the reticular formation of the brain. Then, the potassium chloride injection would induce cardiac arrest and death. Remember, the ionic basis of the resting membrane potential explains why her heart stopped beating. So let's take a closer look. We know that the negative resting potential is a result of selective ionic permeability to potassium and to the metabolic pumps that concentrate potassium inside the cell. However, membrane potential is very sensitive to these changes of extracellular potassium. A tenfold rise in extracellular potassium would severely diminish the resting potential, meaning that the cell cannot rest and it would only be firing action potentials or nerve impulses. Although neurons in the brain are somewhat protected from large changes in potassium, other excitable cells in the body, such as muscle cells, are not. And if you remember, muscle cells, like nerve cells, have an excitable membrane. Without negative resting potentials, cardiac muscle cells can no longer generate the impulses that lead to contraction, and this can cause the heart to immediately stop